Yeah, thank you very much. I also appreciate very much to be invited here. And so, you know, as Reinhard just said, we Europeans are not yet allowed to travel just for fun to the US. And so we have to apply for travel exception. And I wrote to the embassy and I said I want to travel to the US, and they asked me, why do you think it's of national interest that you come to the US? And so I wrote down, I am invited to give a talk about the blood-milk barrier, and 2 days later I had this national interest exception. So I think the blood-milk barrier is of importance. And I'm very happy, as everybody you here in the room, that I'm really here in person and can talk to you. So if you Google blood-milk barrier, immediately you find a YouTube video that explains milk is just a filter, is just filtered blood. And so this would say the blood-milk barrier is just a filter, but you know, of course, it's not, it's much more. So the blood-milk barrier is a border that prevents an unhindered exchange of blood and milk. And we have blood components in milk, but not in different extent. So we have much higher concentrations of milk proteins, lactose, for example, so constituents that are produced in the mammary gland, they stay there. Then we have higher concentrations of potassium, and yeah, as an example. So we have, then we have lower concentrations of plasma proteins, sodium chloride, different electrolytes, citrate, antibodies, and leukocytes. So the blood-milk barrier prevents, obviously, this exchange, this unhindered exchange. So, and this says the blood-milk barrier is semi-permeable. So what is building the blood-milk barrier? Here you can see there are several factors building the blood-milk barrier. So they prevent the exchange here from blood to milk. So first of all, we have the endothelial cells. They are very important, and several authors really investigate the function of these cells in the respect to this barrier function. We have connective tissue in between, and then the basal membrane, and then very important, the milk producing epithelial cells. So actually the blood-milk barrier is built from several structures. So the epithelial cells, they are the most important for this barrier function, because these, you may know, these epithelial cells are very tightly connected and prevent this unhindered exchange. And there are several structures. You find the desmosomes, adherence junctions, and then most important, the tight junctions. So in between connecting these two, the epithelial cells. But these tight junctions are built of several proteins, and they are connected to the cytoskeleton, to actinomyosin filaments, and they also stabilize the cell structure. So, and if we have these tight junctions intact, then the constituents from blood cannot just go. This is a very older picture, but they have to be transported through the cells, and just and cannot, constituents from the blood cannot just travel between the cells to the milk. So we find epithelial cells throughout the whole mammary gland, and usually if you talk about the blood-milk barrier, or many authors talking about the blood-milk barrier, it's meant the area in the alveoli, but you find epithelial cells throughout the whole mammary gland, so in the alveoli, in the milk ducts, and also in the cistern. And if you want to see if the permeability, the function of the blood-milk barrier is everywhere the same, you do not find many publications on this. They are just older ones. I found just older ones from the 70s, from Great Britain, and they say that the permeability is a little different, or the function of the blood-milk barrier is a little different in the different areas. So in the alveolar region, the electrolytes are transported more. So there's a higher transport through the barrier in this area, and less in the milk ducts, and a little higher in the teat cistern. The leukocytes are mainly transfer from blood to milk in the alveoli, and also in the teat, but not so much in the milk ducts. However, water and urea can go in both directions throughout the entire area of the mammary gland. So if we want to characterize the blood-milk barrier permeability, we can use several constituents going back and forth from blood to milk, or also from milk into the blood. And I want to go through the main constituents that we like to measure and to characterize the permeability of the blood-milk barrier. I have here, there's a graph from a study where we opened the blood-milk barrier with an endotoxin, and you can see that several constituents from the blood increase in the milk very fast. And so you can characterize, you can also say how much the permeability is increased. First of all, the leukocytes increase. So maybe the polymorphonuclear cells come into the milk. However, I do not like this marker to be really a marker for the permeability. And you will see later in other experiments I will show you, the results I want to show you. The leukocytes, they come into more into the milk, but maybe this is not really parallel to the opening of the blood-milk barrier. We tested also a lactate, and lactate increases during, if the blood-milk barrier is opened, if the permeability is increased. However, lactate is also released from the cells, and yeah, so, and maybe can travel even through the membranes, so aquaporins in the membranes of the epithelial cells. So lactate is maybe not the best marker, because it's not only traveling from the blood to milk. Serum albumin is a marker that we like to use, because this protein just comes from the blood and has no function in the mammary gland. So the amount of albumin transferred to the milk is really it's really parallel to the permeability of the blood-milk barrier. We also measure sometimes lactate dehydrogenase, and this is a good marker in the beginning of an opening of the blood-milk barrier. However, lactate dehydrogenase is also released from degrading cells. That can be the degrading tissue, also, or the leukocytes that are in the milk. So in the later, after a longer time of blood-milk barrier integrity disturbance, then the LDH can also come from the cells. So it increases, and this is maybe not in parallel with the black milk barrier permeability destruction. IgG, the immunoglobulins, they also increase in milk if the permeability is increased. And here we have to say, so the IgG1 is actively transported during colostrogenesis, but if the black milk barrier is somehow, and if the permeability is decreased, mainly IgG2 comes more into the milk. So this is also a marker, and has likely also immune function. So IgG can opsonize the bacteria. It's that the phagocytes can more easily phagocyte the bacteria. And then, of course, the electrolytes, they are very good markers. So chloride and sodium increase in milk if the blood milk barrier is impaired, and potassium decreases. And then on what I said before, we have markers in blood. Lactose can, if the blood milk barrier is intact, lactose cannot go to the blood through this barrier, and only if the blood milk barrier is impaired, it increases in blood, or you can find lactose in blood, so it's also a good marker. And also the milk protein, or the protein that's produced in the mammary gland, the alpha-lactalbumin, can be measured and is a marker for permeability. So throughout lactation, the permeability of the blood milk barrier changes. We have a high permeability during colostrogenesis, and then throughout lactation, the permeability is very low. So we have an intact blood milk barrier, really to be able to produce milk in the perfect composition that the offspring needs. And then with involution, the permeability increases again, and this is all physiological. And the regulation of this is very complex. So there are many hormones involved. The glucocorticoids, prolactin, and estrogens stabilize the blood milk barrier throughout this lactation, and progesterone increases the permeability again. So this is physiological, but we also see other events when the blood milk barrier is opened, so the permeability increased also during lactation. And for example, we can see a short-term increase of permeability during milking. This was shown, so the lactose increases shortly after milking. However, this decreases again after several minutes. So the opening of the blood milk barrier is very short after milking. And it's discussed that these are the mechanical forces and the oxytocin that is released during milking and milk ejection. It's suggested that this oxytocin, through this contraction of the myoepithelial cells, produce this opening of the tight junctions between the cells, and so the permeability is increased. And then, of course, everybody knows, I think, here in the room, then we have a change in blood milk barrier permeability during inflammation. So inflammation increases the blood milk barrier permeability. Here you can see a study, an older study, after infection with E. coli or Staph aureus. Here was measured the serum albumin in blood, and it increases. And this opening during infection, during inflammation, is pathogen specific. You can see it here already, although here this mastitis was induced with living bacteria, and the E. coli grow even faster. So this could also be a reason. However, E. coli disturbs the blood-milk barrier different than Staph aureus. We like to use, to study the blood-milk barrier integrity, we like to use just the endotoxins, because then you can, we have a short mastitis, but then you can stop it together. You regulate the mastitis, so it's restricted to a special time. So here you can see also serum albumin after LPS challenge increases, so the blood-milk barrier opens during inflammation. To study the pathogen specificity, we used so the cell wall components, LPS, lipopolysaccharide from E. coli, but also from the lipoteroic acid from Staphylococcus aureus. And these pictures, this study was presented before Nabolfa. So if we use these components, these endotoxins in different dosages, so you can induce a different severity of mastitis. So we used dosages that induced a comparable cell count increase. Just to have a a comparable stimulation of the immune system, we used this same increase or comparable increase of somatic milk cells. However, if you have a look at the blood-milk barrier with these two different endotoxins, LPS, LTA, and I showed you here the same samples that had this comparable cell count increase. You can clearly see that after the challenge with LPS from E. coli, you have a much higher increase in IgG2. So the IgG2 just travels to the milk through the barrier, but much less after LTA challenge from Staph aureus. And this is also, and this explains what I said a little earlier, that I don't like so much to use the somatic cell count as a marker for blood-milk barrier opening, because this can be different. So, even though we have the same cell count increase, we have the differences in blood-milk barrier opening, and therefore the transfer of IgG. To study the mechanisms behind this, we used the cell culture model. So we used primary memory epithelial cells, and we always use these cells from different cows in parallel to have the biological replication. We let them grow on transvals, and these cells, they build tight connections. This is sustaining for tight junctions. And if you treat these cells with this LPS and the LTA, and we put the Lucifer yellow, a fluorescent dye on top of this, in the upper chamber of the transval, this fluorescent dye goes through the cells, through the transval, and you find it on the other side. So here in this graph, you see the disappearance of the fluorescent dye in the upper chamber, and you can see, so in black, as if you have no cells, the fluorescence disappears immediately, and with LPS also very fast, but also with a relatively high amount with LTA, the fluorescent dye disappears. And if you do not treat the cells or you treat them with a very low dosage, the fluorescent dye stays in this chamber. And if you look at the next graph, we from these cells, we tested at the end of the experiment, we measured the LDH in the supernatant of these cells. So in this case, the LDH is just released from the cells. Of course, we have no blood in our trans cells, so it's released from the cells. And what you see is we have a strong LDH release from the cells treated with E. coli, but not treated with LTA, although also here in green, this was higher. So here's the LDH from the LTA that you see here, where the fluorescent dye also went through this barrier. So maybe we have here different differences in the mechanism. So we think that we have more cell degradation with the LPS challenge, and maybe for the LTA, it's mainly just the opening of the tight junctions. And this is a picture published many years ago, and they also show that you have during mastitis, you have degradation of the cells of the mammary epithelial cells here. So this is here intact with the control, and here you have it. You see the epithelial cells as an alveolus between at mastitis, and you can see that here the degradation of cells, and here we have hardly any epithelial cells anymore. So mastitis can also, or the blood-milk barrier can also open due to degradation of cells. So this is what I just said, just for better understanding. So if you have an intact blood-milk barrier, so the epithelial barrier, in this case, it's an epithelial barrier. So if you, of course, if you work with epithelial cells in vitro, we have just epithelial cells. So we have little low permeability from these cells. And if the tight junctions are just open, with just the tight junctions open, like after LTA challenge, we have an increased permeability. However, if the cells are also degraded, you have a strong opening of the blood-milk barrier, or the epithelial barrier in this case. So, what are the consequences from if the blood-milk barrier is impaired, like during mastitis, during infection? I showed you have more antibodies and more immune cells in the milk, and this, of course, is essential for the immune response. So this is something good for the immune response, because, as I said, the antibodies can opsonize the bacteria, and the immune cells, of course, can eliminate the bacteria. So this is something we want during mastitis. However, you also have a different milk composition due to this unhindered exchange of blood and milk constituents, and this is something we do not want. So, so maybe the opening of the blood-milk barrier facilitates the cure of the inflammation, and however, the closure of the blood-milk barrier would induce a faster recovery to the normal milk composition. And of course, this arises the question, can we manipulate the blood-milk barrier? And yes, we can. So we can stabilize the milk barrier with anti-inflammatory products. And if you look in the literature, you find several natural anti-inflammatory products that stabilize the blood-milk barrier. We investigated the glucocorticoids and the non-steroidal anti-inflammatory drugs. And I want to show you some results from the treatment with glucocorticoids. So glucocorticoids are already used to reduce the inflammation during mastitis. And we in Europe, we have, or at least in Switzerland, we have also antibiotic injectors for mastitis that contain already prednisolone mostly. You can see it here. I know in the US, I think it's not, you cannot buy them like this yet, but you can, I heard from practitioners that they sometimes use this off-label, also they have this off-label use with dexamethasone, but usually this is applied then systemically, and not in the, intermammarily. However, so we have these injectors, and we know if you treat with this prednisolone, you enhance the cure rate of mastitis, and reduce the inflammation. So what are the mechanisms on these glucocorticoid effects? Glucocorticoids, as you know, everybody knows here in the room, have metabolic effects. They stimulate the gluconeogenesis. They repress the expression of adhesion molecules that would say that it prevents the recruitment of cells into the to the site of inflammation. And glucocorticoids repress the expression of pro-inflammatory cytokines in immune cells, and they stabilize tight junction. I want to show you how this works. So the glucocorticoids bind to the receptor, and they prevent the stimulation of the myosin light chain kinase. And the myosin light chain kinase, MLCK, phosphorylates the actinomyosin filaments. And if this is phosphorylated, they contract and open the tight junction. So if the glucocorticoids inhibit this enzyme, then this tight junction are stabilized. But also the prevention of activation of the NF-kappa B would have the same effect. So if you add the if the prednisolone, the glucocorticoid prednisolone to the into the mammary gland, as we find in these injectors, we did experimentally. So we had again our stimulation with LPS, and if we added in green, we had to the LPS, we added the prednisolone. What you see, you have still an increase of cell count. We couldn't see a difference here, although I just said the recruitment of cells is maybe decreased, but maybe this recruitment is so strong that we cannot see a difference here. However, if we add the prednisolone, we see a lower or limited opening of the blood-milk barrier with our LPS challenge. So in red, you have so this is a serum albumin in milk. In red, you see quarters that just received LPS, and in green, the quarters that received LPS and prednisolone. So the glucocorticoids reduce the opening of the blood-milk barrier, and so maybe you have a faster recovery of milk composition. What we also did, we investigated other anti-inflammatory drugs, the NSAIDs, and they relieve pain and lower the fever and have anti-inflammatory effects. And they are often used in mastitis treatment in addition to antibiotics, usually or mainly for welfare issues, to reduce pain and fever. Two very popular NSAIDs used in mastitis treatment are, for example, the ketoprofen, and it's known the ketoprofen enhances the cure rate and the well-being during mastitis. And another NSAID very popular is the meloxicam, maybe you know. It is also used and recommended for mastitis treatment, and because it reduces the pain and the culling rate, and also increases the fertility after mastitis. So we wanted to know what are the effects on the blood milk barrier if you use these NSAIDs. And first of all, I'll show you how they act. So NSAIDs inhibit the cyclooxygenases, and so these are enzymes important for prostaglandin synthesis. So prostaglandins are important immune factors. They're essential constituents in inflammation and for pain development. And these NSAIDs inhibit the prostaglandin synthesis by inhibiting these enzymes that are necessary for the synthesis. And there are several known from the cyclooxygenases. So the number one is you find in all cells and all the time, and they are these enzymes produce prostaglandins so everywhere in the body and all the time to maintain the homeostasis. And the cyclooxygenase 2 is just induced and increased during inflammation. There are more known in the meanwhile, but these are the two main enzymes involved in the prostaglandin synthesis. So non-steroidal anti-inflammatory drugs inhibit these enzymes, and like for example the ketoprofen I just showed you inhibits both kinds of cyclooxygenases, and in the meanwhile they developed several NSAIDs that inhibit just the COX-2 to prevent side effects, so that it's, the homeostasis is kept the same. And this is an example for this is the meloxicam. However, the NSAIDs also have COX-independent action. For example, they modulate the transcription factor NF-kappa-B, and this would say again we have this mechanism on this cytoskeleton. So we started also again in our cell culture model. So if we treat cells with LPS, our mammary epithelial cells from different cows, and primary epithelial cells from different cows in cell culture, and with LPS you have an increase, so the third bar, and both you have an increase in prostaglandin E2 secretion in the supernatant. However, if you add, that's the last bar, if you add ketoprofen or meloxicam, this prostaglandin E2 secretion is inhibited. So this increase of prostaglandin secretion is inhibited. The cells produce also cytokines when they are challenged with LPS. So here you can see the third bar again is the challenge with just with LPS, and it's TNF-alpha. Here I show you a TNF-alpha expression. However, this is comparable with other results from other cytokines and immune factors. If you add the NSAIDs to this challenge to the cells, this increase of cytokine release expression is prevented. But what is the effect on the barrier function? So for this, we used a special device, a CellScope module, and this device measures the transepithelial resistance in the transvells. So again, we have our cells grown on transvells, and this machine can measure the transepithelial resistance so online, so all the time, in real time. And what we did, we challenged the cells with LPS and treated them also with different NSAIDs. So this is here just an example. We grow the cells until they have a specific transepithelial resistance. That means they build tight junctions, and the current cannot go through. And so you have a normalized, you have a specific resistance. And if you treat the cells, it's here 0.0, then this barrier opens and the resistance is reduced. So here you can see the LPS is the dots, not filled dots. This is an LPS challenge. The barrier opens. However, with the time, the barrier closes again. Here we have a small peak in the control cells. This is why, because when we challenge the cells, we need to change the medium, and we have a short response also on this after challenge, after we change the medium. And for example, here we have a treatment with prednisolone as a control. So you see that the opening is not as far, but the recovery of the barrier is very fast. And so I'm aware you cannot read all this, the ligand of the x-axis. However, I want to show you this, the treatments with different NSAIDs on our cells. And I want you just to get an impression of bar size. So here I show you a challenge with LPS, and in addition, different NSAIDs. So the ketoprofen and also fluniximeglamine. Oh, this is they are both COX-1 and COX-2 inhibitors. Then the meloxicam, you know already, it's only COX-2 inhibitor. Diclofenac, you maybe know from the human medicine. This is a COX-1 and 2 inhibitor, and then celecoxib is specifically developed also to reduce side effects. It's COX-2 inhibitor. And here the graphs, you can see what happens after 5 hours, 10 hours, 15 hours and 20 hours after the challenge of these cells. After we challenged these cells and waited for this time, and we present here the transepithelial resistance. So the first bar is always the control, not treated, and you can see, I think you can see that it stays the same all the time. If we add just LPS, it's the second bar, the resistance is reduced. However, after 20 hours, this is back to the beginning again. And then if you see all the different NSAIDs treated added in addition to the LPS, you can see after 5 hours, the transepithelial resistance is completely down, so the barrier is opened. However, after a time, this recovers. So, and this is NSAID specific. So in some treatments, it goes faster, and with some NSAIDs it takes longer and does not recover. The last bar is our positive control, where we use the glucocorticoid prednisolone. So to to summarize the results from this experiment, that we can see that the NSAID, in addition to the memory challenge, the immune challenge, they do not prevent the opening during the inflammation. However, some of them induce a faster recovery. So they improve the recovery of the barrier integrity. And for this experiment, we used dosages where we compared the recommended dose for use for the cow, that we can compare the treatment. However, we also did the same experiment with the same molar doses, and also there we can see differences in the effects of these different NSAIDs. And what we could not see so in detail, maybe like diclofenac is maybe not a good NSAID because the barrier did not recover. And of course, then we want to see what are the effects in vivo, and we added the NSAIDs intramammarily also, in addition to an LPS challenge. And again, we used the two common NSAIDs that are used during mastitis, is the ketoprophene and the meloxicam. I start with the ketoprophene, and what you see is if you challenge, so these are different quarters, here you see the cell count. If you challenge the quarters just with LPS, you have an increase. And if you add to this challenge the ketoprophene, the increase of somatic cell count is limited. It's lower than just LPS. And if we look at the blood-milk barrier opening, if you here is the serum albumin, and you can see again that in the beginning we have an blood-milk barrier opening, so an increase of serum albumin in milk after we add, after we challenge the quarters with LPS and ketoprophene. However, with the ketoprophene, the barrier recovers faster than with LPS alone. And what is the effect for the IgG content? This is the IgG content in milk is very variable, so this is not so clear, the difference. However, I think also here you can see that with the addition of ketoprophene during this immune challenge, you reduce the transfer of immunoglobulins to the milk. What about the meloxicam? We did the same with the meloxicam. You see the somatic cell count increase. There we could not see a difference between LPS on its own and with the addition of meloxicam. And also that's why, of course, no difference in increase of IgG content in milk. And so we thought maybe we have too strong immune challenge, although we just used already 0.2 microgram LPS per quarter. This is really a relatively low doses, but depends on the LPS you use. This increases the cell count anyway. So we used a little lower, even lower LPS doses. And then we saw a slight difference between the challenge and the challenge just with LPS, and in addition with meloxicam. And but however, here you can see rather an increase in addition to LPS on its own. And again, with the IgG content, this difference is not so clear. However, unfortunately, we don't have longer, so we didn't take longer time samples, so we don't know exactly what is the effect on the recovery after this mastitis. And then, of course, everybody asks what about if you if you give meloxicam systemically, because this is what is usually done orally or systemically. And we also did this. So here we have different cows challenged just in 1 quarter, and just with LPS and without LPS, but both received meloxicam. And so we see a normal increase of somatic cell count comparable to our experiments before, where we have just an LPS challenge without meloxicam challenge. So you cannot see a difference here. And also, of course, then for the IgG content. So in conclusion, for the NSAIDs, so commonly used NSAIDs in mastitis treatment have effects, ketoprofen even more clear effect, and they reduce inflammation, but yeah, they also have an effect on the blood-milk barrier. What you could not see, or what I did not show, we did not show the control. If you add just the NSAID without LPS challenge, we could not see a difference. So this had no effect, although probably if you use a high dosage, then maybe you have tissue irritation. So the ketoprofen increases the blood-milk barrier integrity during mastitis, but not meloxicam. So, but of course, we don't know yet, unfortunately, what the real effect on elimination of pathogens is. So this, we don't have any, we didn't do any experiments on this. So, but if we can close the blood-milk barrier, everybody of course asks if maybe it would be good to open the blood-milk barrier to increase the transfer of immunoglobulins. This could be an advantage for the cure rate, specifically if you have maybe vaccinated cows, then of course you have the antibodies against the mastitis pathogen if you're vaccinated just in the blood and not in the milk. So the antibodies just come to the milk after you have already mastitis. And so then it would be good if we could open the barrier to increase this transfer of antibodies to the milk. And because Sterwang did a longer time ago an experiment on goats, and they used the EGTA to the calcium chelator that takes the calcium away from the tight junction. They need the calcium to be stabilized. And so you can open the tight junctions with the EGTA. However, in cows, it's difficult because it's not very soluble, and you need a very huge volume to inject, and we could not do this in cows. Then we looked at the literature. If you use very high doses of oxytocin, you can also open the blood-milk barrier. So this was done in the 90s. Here it was with 100 units of oxytocin. You can see here, this is the lactose content, so you can open the blood-milk barrier with oxytocin. So we tried this, and here I show you serum albumin. We injected this very high amount, a high dosage of oxytocin, so 100 units. This is really a high dose, and you can see with this injection, we see increased transfer of albumin to the milk. So you see more albumin in milk, so the blood-milk barrier opens after oxytocin injection in this high dosage. Then we did it also with an additional immune challenge with LTA on the top and LPS, and this effect was not very strong. However, we could see a slight further increase of IgG in milk in addition to this challenge with these endotoxins. And then we also did a small field study. We we treated mastitic cows with this high doses of oxytocin. So when the mastitis was detected, the milk sample was taken to analyze the bacteria, but immediately also a high dosage of oxytocin was injected, this 100 units again, and then next day again. And so here you always see the first 3 days after mastitis diagnosis. And there you see, I just present the IgG content because we were interested if we could increase the antibody transfer with this oxytocin and maybe also enhance the cure rate. And here you see the different bacteria that induce the mastitis. We had these results we got later after they were already challenged. So half of the cows were challenged with oxytocin and the others not. And what you can see here, just in the negative quarters. So we took samples from all 4 quarters, and just in the negative quarters, we could see an increase of IgG in response to this high doses of oxytocin, and not the anyway elevated IgG content. This was not changed, and we could not see from this small study. This is for a field study. These are not many cows, so we could not see a better cure rate or faster cure rate in these cows. So we don't know yet if this could be effective. So in summary, for this oxytocin effects, oxytocin can increase the antibody transfer. However, we don't know yet if this can really enhance the cure rate. So I want to give you a conclusion, overall conclusion on the manipulation of the blood-milk barrier. So we can manipulate the permeability of the blood-milk barrier, but nothing is yet established. So the use of glucocorticoids and also this NSAIDs, this is done, but mainly to reduce inflammation, to enhance the cure rate. And nobody, as far as I know, nobody uses these drugs really to manipulate the barrier. And for the NSAIDs, depending on the NSAID and also the dosage, you can increase the recovery of the barrier integrity, but you cannot prevent the opening. So maybe, and ketoprofen you could see can even reduce the somatic cell count increase. So maybe you have effects there. And oxytocin does open the barrier and increases the antibody transfer. However, until now, this is we cannot use this. It's not used for really to manipulate to increase the antibody transfer, because we don't know if this has really an effect on cure rates. But maybe it will be soon. Okay, with this, I want to say thank you, and I told, but I did all these experiments with him, with Rupert Bruckmeier and in his lab, and I will, of course, have to thank all the doctoral students, the master students, and PhD students, and of course, you cannot do experiments like this without the technical help. So I want to thank them and the financial support for most of these studies was from the Swiss National Science Foundation, and the older ones from the German Research Foundation. And for you, I thank you for your attention. And if there are any questions, I try to answer them.